This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 198, recorded on August 29th, 2012. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
How is it there?
It is gorgeous.
Yeah, here too.
It is just absolutely lovely. It's 74 Fahrenheit, a couple of puffy clouds in the sky, sunny. Of course, I'm inside plugged into TWiV, but that's okay.
You could easily go outside.
I could probably do it from outside.
See,
yeah.
I can't easily go outside. So I'm on the 13th floor and
No, that could be hazardous.
couldn't carry my stuff outside.
And if you went outside on the ground floor there, it'd be kind of loud.
It is loud here, but here it's also gorgeous, 24 Celsius.
Yeah.
Also joining us today from North Central Florida, Rich Condit.
Hi, everybody.
How
Hey,
are you?
Rich.
How's it going?
Good. I'm just trying to do a conversion on the temperature here because I just can't, I just can't think it.
Can't
So
do the
we
Celsius?
are 87 Fahrenheit, which means 30 Celsius.
Hey, Rich, what's the permissive temperature for TS mutants?
It's whatever you want it to be. I went, that is a user-defined parameter.
Well, give me one.
Okay.
Give me one.
How about 31 degrees?
All right, see, you can.
okay with that?
You know that in 31 Celsius it's permissive, right? You feel that. You got to just develop the same feeling for everyday living.
I'd
Right.
I'd never done that particular crossover.
Okay.
But I'll work on that. Actually, the non-permissive temperature I can relate to better because when you open the 40-degree incubator, you really get a blast of humidity and temperature. So that's hot.
40 degrees Celsius is your non-permissive, yeah.
Yeah.
Well, and if you walk into the warm room, that's 37.
Holy cow, that's 104 Fahrenheit. Yeah.
Yeah.
No wonder it's hot.
We used to use 39.5.
Well,
I don't know why.
Because the cells die at 40 degrees. So
Half a degree,
we have this.
really?
We're down to 0.2 degree intervals. The actual temperature depends on which shelf in the 40-degree incubator you stick your dishes.
Yeah, right.
The bottom is 39 and a half, the middle is 39.7, and the top is 39.9. And it makes a difference.
All right. Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
Okay, you're
Hey,
okay
Kathy.
to be from Ann Arbor, Michigan, or would you like North Central America, or
Ann Arbor is just fine.
where are you in Michigan? Would you be North Central Michigan?
No, southeast.
Yeah,
Southeast.
I could never get you and Gainesville straight. So yeah, it'll be Ann Arbor.
Yeah.
You have nice weather today?
Just like you, 76 Celsius, puffy clouds.
No, it can't be 76 Celsius.
Sorry, Fahrenheit,
Fahrenheit.
24 Celsius.
Yeah,
Sorry.
Yeah, 76 Celsius is
After all
the
that.
non-permissive temperature.
That would, yeah. Sorry.
Is it really dry out there?
We've had our drought, but we've also had some rain. We had rain earlier this week, Monday.
Uh-huh.
Yeah.
Because you guys have a West Nile thing out there, too, I know. I was looking
Yeah.
today, a couple of deaths.
Mm-hmm.
And Dixon tells me that the hurricane is going to take care of all that. Of course, it won't take care of it up by you because it's not going to get
No.
anywhere near you.
Right.
All right, here we are to do a TWiV. No Dixon, he's out pulling weeds, or weed whacking as he calls it.
Wait, I didn't think you had to do that in the vertical farm.
Very good. I went to his fishing club in Pennsylvania. Listen to this. I said, where are you going? He said, I have to go get rid of the invasive weeds. I said, invasive according to who?
Yeah, exactly.
Right.
Didn't we have that discussion last time?
We
Or-
did.
Invasive. Just let them grow, right?
Well, a weed is any plant that you don't want growing in that spot.
Right. So
Yeah.
invasive weeds, in fact, is a redundancy.
It's redundant.
Yeah,
Yeah.
weed is another word that is a weed to us, but not to the weed, right?
Right.
I bet you plants don't call weeds weeds, right?
I don't know. Some plants probably don't want those other plants growing there.
Yeah.
They probably call them weeds.
To a plant, probably every other plant is a weed.
Yeah.
Okay. This week in weeds, we have
We're doing the biopsychology of plants now.
yeah. You know, I did this interview with Philip Marcus for the last TWiV. It went up last week, and it's a part where he was recounting he knew Leo Szilard, the famous physicist who became a phage biologist. Mm-hmm. And he was explaining to Szilard how he couldn't clone HeLa cells. When you put single HeLa cells in plates or whatever, they would die. So Szilard thought about it, and he said to him, "You have to fool them into thinking they're with other cells." And Marcus said to himself, "So here's a physicist going into biopsychology now."
That made me laugh out loud. That was good.
That book, Making of the Atomic Bomb, which was my very first TWiV pick, starts with a story about Szilard.
Yeah.
Having the insight that the new understanding of atomic structure could lead to a bomb. That guy must have been just amazing.
He's all over the place.
Yeah.
I was out at Cold Spring Harbor on Friday, and I mean, you don't escape his influence. There's all kinds of artifacts all over about the place, and people are talking about him. He's a very old microbiologist and now eukaryotic biologist, Wacław Szybalski.
Yes.
You know that name?
Mm-hmm.
Yes, I do. He was the mentor of a guy named Bill Summers, who was on my thesis committee at Yale.
He's not too far from you, Kathy. He's up in Madison.
Okay.
All right, so you don't think it's close, right?
All those Midwestern states are kind of in the same place to a New Yorker. So.
Well, isn't it like an hour from you? Or is that Chicago I'm thinking about?
More like 8, and Chicago is more like 4. Oh, so Madison's probably more like 6, yeah.
I think you guys are right next to each other. Well, that's the New Yorker view of the world.
Yeah, there you go.
Okay. Anyway, now I've forgotten why I brought all this up.
You were talking about Cybulski and Cold Spring Harbor.
Yeah, so Cybulski was telling stories about Szilard, too. All over the place out there. Hey, I saw Hershey's blender out there. They have
kidding.
They have it in a glass case in the library. Yeah.
Cool.
And they have a letter from Hershey accepting the job offer at Cold Spring Harbor. And he says, "I don't think I can accept less than my current salary, which is $7,000 a year."
Amazing.
Anyway, we have 2 papers to do today, but there are 2 quick follow-ups from recent TWiVs. One is from Ed Millson, who writes, "Great TWiV 197. It is really nice to learn how science was. Fascinating to see how laborious it was to do things that nowadays are only kitology. Keep up the great podcast." That's the Philip Marcus episode. I really enjoyed talking to him.
Yeah, I listened to it. It sounded like a good time.
He was fun, and it's neat. I loved his thought process. What was going on back then?
I like all the images you put up on the website too.
I agree.
The things he talked about. Yeah,
he gave a lot of those to me. It was really nice. I think that stuff is important to get down. You know,
I agree.
Absolutely.
So it's there forever. I mean, these big people in science are not going to be around forever, and it's good to have it for future people who are thinking about going into science. They can listen to this stuff. You know, I just think they would do it rather than reading something, right?
Mm-hmm.
Right.
Yeah.
So, the next one is from Coin, who writes, "After watching Mark's science pick from TWiV 196, a YouTube channel dedicated to interesting and entertaining videos shot with a slow-mo camera, I felt I had to share a link that I came across the other day." Puts a link to a YouTube video. While standard slow-motion cameras recorded about 5,000 to 10,000 frames per second, this technology, which is dubbed femto photography, developed at MIT, captures light at 1 trillion frames per second. Because of the incredibly small amount of light that is captured with each frame, The videos they show are computationally reconstructed from millions and millions of events with very clever synchronization. In one resulting video, we're able to watch a 1 millimeter long packet of photons travel and scatter as it hits a surface. A proof of concept demonstration also shows that the technology can be used to see around walls through light, reflection, and more clever computation, which could be beneficial in biomedical imaging. Thank you for the podcast. I've been interested in viruses and working in virology labs for several years now, and TWiV always gives me something new to think about. I like that his name is Coyne, his first name.
Yeah.
And remember, Carolyn Coyne was on TWiV not too long ago. It's amazing stuff. Trillion
Yeah, it
frames
is
per
amazing
second.
stuff.
Yeah, I saw that when it came out. It was slow motion photography of light. It's just
Good stuff. Science and technology. All right, let's move on to the literature, as Mark Crislip would say. Anybody ever listen to Mark Crislip?
Mm-hmm. Did once,
the
at least. Yeah.
Emergence of fatal avian influenza in New England's harbor seals, published in MBio. And I've had this on the agenda for a couple of weeks. It keeps getting bumped. But this is from the Lipkin Laboratory here at Columbia in collaboration with the New England Aquarium in Boston and a few other places. National Oceanic and Atmospheric Administration. Hey, that's where I go to see the weather online. NOAA.
the National Wildlife Health Center in Madison, Wisconsin, right up where you are, right, Kathy?
Well, it's about 8 hours away.
6 to 8 hours away.
6 to 8 hours, depending on how fast you drive.
Right.
Now,
and what the traffic is like in Chicago.
You have to go through Chicago to get there? Yeah.
Yeah.
This is about seals who get flu, and that happens now and then. Although I remember when it first happened, which I think was in 1979, it was kind of a surprise. Well, I shouldn't say that. It was first detected.
First observed.
But I bet it happened long before, right?
Sure.
We just didn't pick it up. Nobody looked at seals. But, you know, when a bunch of them wash up dead, then it gets some attention, right?
Yeah.
So according to this paper, there have been a number of outbreaks from 79 to 80 and 91 to 92. And they involve various influenza viruses, H7N7, H4N5, H3N3. These are combinations we never talk about here on TWiV. But
I was thinking that it might be good to, in case we have really new listeners, to remind them what the H and the N stand for. And they're abbreviations for surface proteins that are found on the surface of the virus particle. The flu viruses are enveloped, so they have a lipid bar with proteins sticking out. And H is hemagglutinin and N is neuraminidase.
Right.
Okay, Kathy, how many H's are there?
Oh, I think there's up to, 9, 15, something like that.
I know there's at least 13. I think there's probably 15.
I think there's 17, including the new one from bats in Guatemala recently.
Ah.
And there's at least 8 N's. I guess that's obvious from the paper, but there's probably more, right?
There are 9 N's.
Okay. And these hang out mostly in birds.
Right.
Yeah, every known flu is in birds, yeah. Only 3 of them are in people. H1, 2, and 3. Anyway, this paper investigates one of these outbreaks. So it was in September 2011. Do you remember this, Alan? Do you get this news up in England?
I remember some investigation of the harbor seal deaths, and they were washing up, and people didn't know why, and there were a lot of theories floating around about what was going on.
Floating around, is that a pun?
Yes. Well, no, actually, it's literal. I think they were washing up on shore.
Yeah, 162 seals washed up on shore.
And I don't mean taking showers.
Dead or moribund. So moribund is very sick, right?
Just about dead, yeah.
This number is 4 times the expected mortality. So seals do turn over. They have a half-life.
Sure, yes.
And these were pretty young, less than 6 months old.
Every seal gets broken eventually.
Ooh, wow. So they wanted to know what was with these seals. So Ian Lipman got samples from 5 of these seals, 5 of these 162 seals.
Now, he says 5 animals were submitted for analysis.
Yeah, that's fine.
I don't know. I would just ask for tissues, but...
Yeah, I'm sure he just got tissue, right?
Yeah, well, maybe his collaborators, maybe they FedExed them out to Madison and took them apart there or something.
So they got tissues from a variety of tissues. Sorry, they got samples of lung, trachea, liver, kidney, lymph nodes, spleen, skin, oral mucosa. Made nucleic acid and did PCR, and they found H3N8 influenza.
Right. So they did PCR for just a whole bunch of families of viruses.
Right.
Not knowing what they might find,
including radicchioviruses.
Right.
No, I'm just kidding. That's no radicchiovirus. That's a vegetable.
Yes.
And found H3N8.
There might be a radicchiovirus.
I'm sure there is, actually.
Are they related to arugula viruses?
Sure. They're segmented. That's what happens when you do TWiV on a Wednesday.
Yes.
I'm sorry, everyone. I'll calm down. So they got the sequences. And the cool thing is, or at least what I thought was cool, they made extracts from these tissues and inoculated them into eggs, and they got viruses too, and showed that they were in fact H3N8.
Yeah.
So not just sequence, but actual virus, which is good, because sometimes in these discovery exercises, they only get sequence.
And they recovered consistent sequence from all the individual animals that they looked at.
So that brings up the question. So in these 5, you get this one virus. It's the same in all the animals, or more or less the same. Did it kill all 162 seals?
Well, yeah, I had this same issue, because somewhere down here, after they do this pathology and they show that they can see the virus in the lungs and et cetera, they draw the conclusion that this is the virus that killed these seals.
Right.
Right.
And I kind of sat up at that, and I thought, well,
it's a little strong.
Really, it's a little strong. But on the other hand, I'm going to have to find that conclusion again. But
at the same time, you're talking about 162 dead seals. You sampled 5 of them, and 5 out of 5 were positive for this one particular virus, and not for any other virus.
Yeah.
It's strong circumstantial evidence that this is what's causing the problem.
Rich, I think where you might be talking about is just on the first page, about 6 lines from the bottom on the left, where it says the H was responsible for the observed clinical and pathological signs in these animals.
Right.
Yes.
Here we demonstrate that.
Yeah. So that's a little softer conclusion, but still.
Yeah.
And I thought about it for a while, and I decided, I'm not going to be picky here.
It's probably not something
strong
else.
evidence that
this
virus killed these seals.
These 5 anyway.
Yeah.
But the other, you don't know about the under 162.
You don't know about
because some of them may be natural deaths, right?
But they're pointing out, this is higher than a normal mortality, and the ones presumably they looked at a fair number of these, and it wasn't the usual sorts of causes. It wasn't that they were malnourished. A large number of them were less than 6 months old. So there are some demographic factors in this population of dead seals that it all points toward it being a single cause.
Yeah.
But yeah,
I think they ought to put them in ferrets, these viruses.
clearly.
That had to come up. I knew that was going to come up.
Well, actually, I'm serious, because they want to know if they're transmissible. But we'll get to that. So this is called A. Harbor Seal, Massachusetts 1, 2011. That's the way you name influenza viruses. The A is type A, the species you get it from, where it comes from, an isolate number, and then the year.
Right. So H3N8 is defining the serotype of the virus, and then this more specific A. Harbor Seal, Massachusetts 1, 2011 is the rest of the designation. It's not just any H3N8, it's this specific one.
Right. They did some hybridization of sections from these seals to show that, in fact, there was virus in there. They did immunohistochemistry and nucleic acid hybridization. So the virus is in these animals. So I guess that's good evidence that those 5 animals were infected with virus, right?
Yeah.
Mhm.
And then they sequenced the entire genome so that they could interrogate it.
Right.
Yeah.
Anybody like the word interrogate?
Actually,
Not in this context.
I have begun. I must say that I used it a couple of times today in a qualifying exam.
Rich.
It's useful, Vincent. I'm sorry. It's
I
useful.
But you should have been saying, "Let me interrogate you about this."
Yes.
The only way you could use it.
They say here,
I was interrogated in my qualifying exam, but it was quite different.
the seal H3N8 genome was interrogated for any genetic features that might contribute to enhanced transmissibility and virulence.
So they showed it a rack of restriction enzymes, and they said, tell us where you've been.
You know, I view this as a meme, okay? A couple of years ago, somebody used this in a paper to say, I'm going to interrogate this library or this microarray, and now it's stuck and everybody's picking it up.
Yep.
It's not the right use of the word.
And I have a few problems with it. It's a new use for an established word. It's also, it encourages use of passive voice. So the genome was interrogated, not we analyzed the genome, which is actually what happened.
Yeah, in my day, we
We've got
used to say
perfectly good words for this.
we searched the sequence for this or that. We searched or we screened. What's wrong with those ways of saying it? I don't get it. I mean,
This is the hip way to say it, I guess.
It's hip, yeah. It's a meme. Okay. I have no objection to the English language evolving, but I don't see why you need to make a new word. Like reverse genetics, that's another one.
Yeah.
Okay, so they looked at the sequence. They did all 8 segments, and they found, first of all, which is important, the closest virus was one that had been previously identified in North American waterfowl, right? So the implication is that this virus came from some aquatic bird and went into the seals. Everybody okay with that?
Yeah.
It was foul play, as usual. Yes.
In fact, it's the closest avian relative is from a teal, a blue-winged teal, and it was isolated in Ohio in 2002.
Them's good eating.
Them's good eating. That's snakes, isn't that snakes?
No, the teal are really good eating. I'm not kidding about this one.
Okay.
Are you serious? You've eaten teal?
Oh, yeah.
In what restaurant do you eat teal?
When you're a little boy and your father's a hunter, you go out and you shoot these things. You
Really?
bring them home and you grill them.
Wow.
And drizzle them with lemon butter, and it's just to die for.
Well, it's a duck, isn't it?
It is a duck.
Yeah.
I bet you have antibodies to some of these avian viruses.
Could be.
You know? And you should find out, and you should interrogate your serum and find out, because then you could say, look, I have antibodies to H5N1 and I'm fine.
There you go.
All right. So the seal virus and highly related avian H3N8s differ by 37 amino acid changes. So they want to do this analysis now to see how close the viruses are, right?
Well, and teal and seal differ by only a single change.
Teal and seal.
Wow.
Are you annoyed with us, Kathy? I know
You don't have to
Entertained.
answer that.
You're usually much more serious than we are. I'm sorry, but maybe you'll be infected as TWiV goes on. You get the TWiV sense of humor, which is patented by Alan. So we have 2 particular changes. Oh wait, sorry, 2 mutations shared by seal and other H3N8s that have been isolated from mammals like dogs and horses. And there's a change in the PB2 protein, which is one of the subunits of the RNA polymerase. It's 701N. And also a change in the hemagglutinin at 260. All right. And then there are a bunch of amino acid changes shared by the seal and human H3N2 viruses. So they also compared this H3N8 to H3N2, which is a human virus, to see how similar they are. And there are a number of mutations common between the H3N8 in seals and the H3N2 in people. So the idea is that, can we predict that this H3N2 might go into people, I suppose, right?
Right.
Yeah. Well, importantly, this is a classic example to start with of this thing jumping species from birds into seals and killing them.
Right.
Now, actually, we don't know if the seals gave it to each other.
Right, we do not know that.
Right.
Yeah. And in fact, they are hedging that at the end. They don't know if it's transmitted among seals, which is why I brought it up that we should put this in ferrets to see if it transmits among ferrets, right?
Are ferrets a good model for seals?
No, they're a good model for ferrets.
Right, but they're the best model we have, right?
I seem to remember these statements.
Yeah, something like that.
But I would predict this virus would not transmit among ferrets because it doesn't have any of the amino acid changes that Fouchier and Kawaoka had identified.
I wonder if those experiments are going on.
I don't think you can do transmission stuff. Well, that's H5N1, yeah. So the moratorium is still on, as you may know, right?
Mm-hmm.
But it's only for H5N1.
Yeah, that's right. So they could do this in ferrets. The PB1 change is interesting. Sorry, there is a PB1 amino acid change in the seal that's similar. That's the same as the one in H3N2, and they think these are adaptive, representing adaptive changes from the bird to mammals. It's known that these PBs have to change to get better replication in mammalian cells versus avian cells. So that could represent adaptation.
And that's the reason for calling them host range determinants.
Yeah,
right? For PB2 anyway.
Yeah, that's right.
And host range comes up in the second paper that we're going to talk about. So just wanted to make that point that host is talking about what kind of species a particular virus can grow in or what kind of cells it can grow on. So PB2 is important for host range.
And that it can involve things that go way beyond what cells it can attach to or something like that. In this case, the polymerase, which is actually involved in the nuts and bolts of replication, is a determinant in host range. That's not obvious.
Right.
Right.
So here, I'm going to jump to later on just to finish with this PB2 business. So PB2 is a determinant of host range. There's an aspartic acid in the bird strains, which gets changed to an asparagine at 701. All right, and that's been known for a while that D701N influences the pathogenicity in mammals. Okay, so the virus as it comes out of birds is not as pathogenic in animal models if you don't make that change, and that's one of the changes introduced into the H5N1 that was important for it to be transmitted. So the seal virus has D701N. All right, so bird viruses don't have that. The seal virus does. So the implication is that it was selected for maybe by transmission, right? But we don't know. We don't know. The other thing they look at is there are these amino acid changes in the HA, and they do some modeling and predict that these are going to affect the kind of sialic acids that are recognized. So to remind everyone, the avian strains prefer alpha-2,3 linked sialic acids.
These are sugars on the surfaces of the cell that the virus is going to bind to.
Mm-hmm.
Yeah, flu has a sweet tooth, right?
Yes.
Right. You would have said that if I hadn't, right?
I would have gotten to it, yeah.
Sialic acid, alpha-2,6 linked is the linkage preferred by mammalian strains. So, in fact, they do an experiment to prove that the seal virus is able to recognize alpha-2,6 sialic acids. It can use both alpha-2,3 and alpha-2,6, which is what happens when avian viruses get adapted to mammals. And you remember, again, the H5N1 ferret story. That's one of the changes they introduced to change the receptor recognition from alpha-2,3 to alpha-2,6.
And did I understand correctly from this that depending on where you're looking, the seal lung contains both alpha-2,3 and alpha-2,6?
That's what I got, yeah.
So that makes it, an optimal sort of mixing vessel, where you could take a bird virus that prefers one and get it into the it might have a chance in the lungs, and any variants that came up that would bind the other receptor could be amplified. At least that's the way I look at it.
So they looked at the pulmonary distribution, experimentally using lectins that either bind to alpha-2,6 or alpha-2,3, and you can detect the lectin binding by fluorescence or some other marker. And the alpha-2,6 was throughout the lung of the seal, but alpha-2,3 was mostly in the bronchioles, way down below. And they say the diffuse infection seen in the lung of these animals is not consistent with the alpha-2,3 localization. It's more like the alpha-2,6. So these viruses are probably infecting via alpha-2,6 in the seals as well. Yeah.
And this is something that's very strongly parallel to the situation with bird flu going into humans, right?
Exactly.
Because the alpha-2,3 receptors are deep down. The bird virus can get into people occasionally if it gets down deep enough and can infect those cells, but it doesn't transmit as well. It doesn't really appear to transmit human to human at all efficiently if it can't get up into the upper part of the lungs.
Right. That's where it has to replicate to be transmitted, right?
Right.
So this is, you can see now why they're doing this work, because it has parallels between H5 going into people and some other avian virus, because we shouldn't, I guess, focus just on H5. There are other avian viruses out there that could go into mammals.
It's curious to me that over millennia, I guess, of all of these different hemagglutinin and neuraminidase types, that only a limited number have wound up in people.
Yeah, it's true.
Yeah.
I would say that considering all the viruses on the planet, that very few have come into us at all.
That's true. Right? Zoonosis is not a real, it happens a lot, but considering the larger picture, it's pretty rare.
Yeah, most of these types of cross-species transfers, I think across infectious disease in general, they end up being failures for the infection.
That's right.
Right.
They may kill the first individual they infect, but then that's the end of the story in many cases.
So that is about it. They say this could be a zoonotic threat because it has these HA and PB2 changes that could be prerequisites for transmitting among people. And we should pay attention to this. They say, well, we don't know what these changes... They say further studies will be required to establish the significance of the substitutions in this genome. So I assume they're going to have to use some model to look at that, and not seals, because nobody uses seals as experimental animals, right?
No, it's difficult to keep in the lab.
And so they will probably have to use ferrets or something else.
Yeah, and the seal population, as a coda to this, the seal population in Massachusetts Bay and along the coast here is still extremely high. We had a booming population of them this year, apparently, which was the reason there were all those sharks sighted off the coast, because they come in to feed off the seals.
Oh.
That's right, yeah. So it says they must be considered a significant threat to wildlife and public health, these viruses. So what do we do? What should we do?
Watch them.
Watch them,
this
exactly.
has been going on forever.
Yeah.
Right.
You mean we should sample birds and seals and just pay attention to what's there?
Sure, do exactly this kind of stuff.
Yeah.
Right.
And anytime you see an unusual pattern of deaths, like in this case, you have 4 times the normal mortality from the population of seals, you delve into it, see what's going on.
Okay.
And you accumulate a bunch of data over time that gives you clues into what makes this happen, what influences transmission and pathogenesis, and
And it may be that this sort of thing just happens every few years, and we're just now noticing.
Yeah.
Right.
Or this may be freakish. We don't know.
One thing that they do talk about when they're talking about Table 2 is where they're specifically trying to talk about transmission from seal to seal. And they say analysis of the hemagglutinin sequences over the course of the outbreak show the introduction and maintenance of 2 nucleotide polymorphisms. And while this is insufficient to convincingly demonstrate seal-to-seal transmission, it leads us to postulate that mammalian spread might have already occurred. And I guess I want to take a little bit of issue with this. This is another place where I thought the statement was maybe overly strong, because they say over the course of the outbreak, but in fact it looks like the 5 seals that they have are from the 28th of September till October 3rd. And
Right.
and I don't think that the data that they show in Table 2 is particularly convincing, given the small sample size that we're seeing seal-to-seal transmission. And then yet a little bit later they say, this is the natural epizootic emergence of a pathogenic virus that can transmit between mammals. So I guess I would have liked to have seen them be a little more cautious.
Yeah. In fact, Table 2 is just 3 animals. Right.
278,
278, 286, and 295.
Well, the last one I think is 294. I think the table that got omitted because they talk about it in the figure legend.
Oh, okay.
So where is this virus now? It probably can't go back into birds, right? Because it's lost properties that let it grow well there. So it's either in the seals or it's gone, right?
Right.
So it seems to me you should go out and sample seals to see if it's still there. Because it could be if there's the next seal outbreak, it's a different virus, right?
Right.
Yeah, I would hope that they're sampling some seals this season, maybe from ones that die naturally because a certain number do,
Yeah.
or pieces that the sharks missed or something.
Right.
However one goes and samples seals, I'm not sure.
Well, you can't just walk up to them and ask for a nasal swab.
No, especially if you want kidney and trachea.
They want you have to get moribund or dead ones. So
Yeah.
pick them up off the beach, but they don't cooperate. Yeah. Okay.
There's an example of this. In the introduction of this, they talk about outbreaks in the past and name several of them. There's one of these that showed up in an early edition of Fields Virology that I use in my flu lecture from 1982, where in fact they're able to show that the virus that is generated in the seals is a reassortant between 2 pre-existing fowl viruses. So the seals served as a mixing vessel in that particular case, and came up with something that killed them.
Right.
So,
All right. Let us move on to our next paper. This one was sent in by a listener, and his name was Patrick, and I will read his letter later. But this is a paper from Harmit Malik's lab, who has been on TWiV before. It's called Poxviruses Deploy Genomic Accordions to Adapt Rapidly Against Host Antiviral Defenses. First author is Nels Aldi, Child, Ike Bush, Kitzman, Rogers, Shendur, Gabal, and Malik. And it's published in Cell. And I'll bet you're really excited about this, Rich.
I'm liking it.
It's poxviruses, man.
And genomic accordions. What the heck are those?
Yeah.
Squeeze box.
LD is now at the University of Department
Utah?
of Human Genetics, University of Utah.
I met him at
And just-
ASB, and I didn't realize until this morning that was the person that I met. But
Yeah, that happens.
yeah, when I saw his video, so we were going to talk about the fact that he's. So you've heard about visual abstracts for papers, and now there's such a thing in Cell, evidently a video abstract.
Yeah,
And-
I was unfamiliar with this, but this was amazing.
Yeah.
Yes.
What is it that they call it?
A paper
Paper
flick.
Paper flick.
Yeah, a paper flick, a flick about the paper. Yeah. And when I saw the video, I thought, oh, I think I met him. So, yeah, there's
Yeah.
people
The
behind the
video is
scenes.
really entertaining. It's very good.
Yes.
Yeah, check it out. It's in the abstract area, so you can get at it without having to subscribe, I think, right?
I think so.
Yes, you can.
I like it because
Mhm.
it shows these guys aren't all running around in white coats with pocket protectors and stuff like that. They have a sense of humor.
Right.
And he asked the question, something like, how does it do that? And they look at the camera and shrug their shoulders.
Yes.
I had heard this from Harmit when I visited UW last year. He told me this story.
I think he talked about this at TWiV year before last.
You think?
I think. I seem to recall that was part of he gave a plenary session talk. And I know he talked, well, I believe he talked about this work.
I want to just say one thing, and then I want to let you take it, okay, Rich?
Okay.
I like this sentence, the second sentence of the paper. Many RNA viruses counter host immunity with high mutation rates coupled with short generation times and large effective population sizes, allowing rapid exploration of mutational space for adaptation. So I think that's beautiful. And they're contrasting that with poxviruses, DNA viruses that have a lower mutation rate,
right,
smaller mutational space. And that's the subject of this paper. I like this, exploring mutational space. Is that like the thing that went to Mars?
It's a little
mutational
different.
Space, the final.
Oh, not outer space.
No.
Okay, go ahead, Rich.
So I guess the basic thesis here is to understand how viruses evolve, and in particular, in a situation as Vincent just set it up, RNA viruses, because their replication enzymes don't proofread, can mutate very rapidly, and that gives them an advantage in terms of adaptation. DNA virus polymerases do proofread. They have a much lower mutation rate, and so the question is, with such stability in their genomes, from a mutational standpoint or a point mutation standpoint, how do they manage to adapt fast? And so the idea was to set up a situation where you force a particular virus to undergo mutation to adapt to a situation, and then ask specifically from a genetic point of view, how does it do that? And they chose as a model system the poxvirus vaccinia. And just to remind people, this is a large double-stranded DNA-containing virus, and by large I mean 200 kilobases, so we got enough for about 200 genes. It's unusual in that, although this isn't all that relevant to this paper, it is an interesting sidelight. It's unusual amongst DNA viruses in that the whole replication cycle takes place in the cell cytoplasm. I would imagine that this same sort of thing could apply to DNA viruses that replicate in the nucleus. I don't think the cytoplasm has anything to do with it. There are other features of the biology of the virus that I think facilitate this, what we're going to discuss here, and we can get to that.
Well, I think they even mentioned some other DNA viruses that might do some similar stuff in the
there's certainly
discussion.
There's other DNA viruses that have evidence of multi-gene families in them.
Right.
Okay. So, about half of the poxvirus genome probably is devoted to dealing with these innate and mostly the innate immune response to viruses. So there's a tug of war going on between the cells and the viruses, which is a common theme on TWiV, and the viruses are fighting back against various cellular and organismal immune defenses. One cellular innate immune mechanism is a protein called PKR, for protein kinase R. And it does lots of things, but we'll just highlight one of them for the purposes of this paper. This is a cellular protein that normally is just kind of hanging out, but it is activated by double-stranded RNA. Double-stranded RNA being something that is typically made during virus infections. Now, you might ask, how do poxviruses do that? Well, late during infection, RNA viruses obviously have double-stranded intermediates in their replication. Poxviruses late during infection have some late genes that have converging promoters and really long messenger RNAs, so there's an opportunity there to make double-stranded RNA, which they do, and the cells see this and go, oops, this is not good. So they have cells have evolved mechanisms for sensing double-stranded RNA, and that to them means danger, and they respond to that. One of these sensing mechanisms is protein kinase R. It is a double-stranded RNA activated kinase that, when activated, does a number of things. Among other things, tells the cells to kill themselves, fall on the grenade, as it were, to save their buddies. But one of the things that it does is that PKR does is to phosphorylate a translation initiation factor called EIF2 alpha that has the effect then downstream of shutting off protein synthesis. So that's going to mean both cellular and viral protein synthesis, but at this point, this cell's cooked anyway. So once again, it's falling on the grenade, but at least you can shut it down and prevent this virus from completing its replication cycle. So vaccinia virus actually makes a couple of different proteins to circumvent PKR. One of them is called E3L, and the other is called K3L.
These are great names.
Yeah. They're not real descriptive. If you hang out for long enough, of course, they acquire meaning. These are all just named for their position on the genome. E3L is a protein that binds double-stranded RNA and sequesters it so that the cells don't know it's there. K3L is a really interesting protein. That is a protein that is homologous to EIF3 alpha. So it looks like the target, it looks like the protein, the initiation factor, although it doesn't function in that fashion. And so the PKR sees this thing and binds to it. It's a competitive inhibitor, basically, of PKR. Binds to it, it's confused, and it'll actually phosphorylate it, I think, but that doesn't really matter. It binds up K3L, so it can't do its normal job of phosphorylating EIF2 alpha. So K3L, by homology to EIF2 alpha, is a competitive inhibitor of EIF2 alpha. So what these guys do is they decide to set up a situation where the virus is crippled in its defenses against PKR. And to do that, what they do is take a virus and delete the E3L gene. So this double-stranded RNA binding protein is now gone. And it was known previously that if you do that, you wind up with a virus that has a host range. It will grow in some cells, but not in others. I guess I don't know if the mechanism of the cells that are permissive is entirely understood, but clearly they must not be as sensitive to the double-stranded RNA issues. But in the cells that these guys are going to use, the E3L mutant cannot grow because PKR is activated. So then they ask, okay, so can we make this virus now, will this virus adapt to this situation and grow on these cells where PKR is winning the battle? And in order to ask this question, they take these viruses that lack E3L and they infect cells at a fairly low multiplicity. It's like 0.1 or 0.01 plaque-forming units per cell, and they allow time to pass, and not much happens because the virus, it's non-permissive. But what they do is they harvest that stuff, they get a little bit of virus out of it, and they do another similar infection, and they do it again and again, and they serially pass the virus, and lo and behold, after several passages, what they find is that viruses are now growing on these cells that are otherwise non-permissive.
And I like how, as they're doing this serial passage, they remove aliquots and reserve them for what they call the fossil record.
Yes, right. That's good. So they keep samples all the way along.
I got a freezer full of fossil records, Alan.
Yes. Yeah, so they're tracking evolution. Why wouldn't it be a fossil record? Yeah.
Never looked at it that way.
So they wind up with viruses that don't do quite as well as a wild-type virus would, but are certainly much, much, much better than they started with. They're growing not too bad.
Rich, these are HeLa cells, right?
Mm-hmm.
If you take the parental vaccinia with both genes intact, K3L and E3L, does it grow much better than the deletion mutant? Because I don't see it here.
They have a in this figure 1C, they have a little dotted line to show, I think, what with a wild WT next to it. And I think that's what they expect wild type to do.
So it does fine, doesn't it?
It does. These do pretty well. It looks like they're reduced a little bit, but not even a log.
Yeah,
just a few fold.
Do you use HeLa to grow vaccinia, or do you use some other cell?
One can. I use monkey BSE 40 cells. People typically use mostly monkey cells, but actually we do use HeLas. We use spinner cultures of HeLas for mass production.
Mm-hmm. Okay.
We started off using BSE 40s because they're alleged to grow well at a high temperature. Back to the beginning of our conversation, 40 degrees. That's what the 40 means. Of course, we haven't kept that pressure on. So
I knew there was a reason I was mentioning that.
Go. So now they analyze these things to try and understand genetically why it is that they can now grow under these conditions. And they do it by isolating bulk viral genomic DNA from these cultures. So if there's heterogeneity in these populations, they'll get everything out, and sequencing it in bulk, which is a really interesting approach to this. And they just compile all those sequences together, and what they find, this is fascinating, what they find is that the number of reads that they get that correlate to the K3L gene is in vast excess to the number of reads that they get for all other genes. And the implication is that there are extra copies of the K3L gene. And it's interesting because they have sort of an internal control because they don't get any reads for E3L, which you would expect because that's deleted. And so then they have a closer look using more ancient molecular biological techniques like southern blots and stuff. And they find, looking at their fossil record and the current viruses, that yes, lo and behold, these viruses contain a whole slug of tandem repeats of the K3L gene. So the way, apparently, at least initially, these viruses have overcome PKR is by attrition, I guess, is to make a whole slug more K3L so it does better at trashing or competing out the PKR in these cells and allows these viruses to grow. Now
they actually validate that by knocking down or overexpression K3L.
Yes.
Yeah, so
right. They validate it in a couple of ways. So they use siRNA to knock down the K3L expression in those amplified viruses, and now they can no longer grow. It's interesting to consider how this process happens. And I always have to draw this out to make it make sense to me, but it's basically an unequal crossover between two genomes. And they actually show some of these junctions where you can cross over unequally between the upstream gene on one genome and the downstream gene on another genome. And what that generates out the other end is a duplication of the K3L gene, which was in between, and a bogus gene that's a hybrid between the two where the illegitimate crossover took place. But that's okay because those two genes still exist in their intact form in the recombinant virus. And then once you've done that duplication, then you can actually do homologous crossovers through the duplicated K3L, and the amplification is much easier. So these guys have amplified K3L genes with some other sort of junk genes that are bogus recombinants in between, but that's okay because the important thing is they got extra copies of K3L.
So the virus normally has two approaches for dealing with this PKR. It hides the double-stranded RNA with E3L, and it also kind of tries to cover up the PKR with K3L.
Right.
And then you take away the E3L. Now it can't hide the double-stranded RNA,
right.
But the solution is to pump out lots and lots of K3L, and it can still mask the PKR and muffle it.
Right.
Yeah, in this case, the solution is not dilution.
Right.
Right. So now the other thing that they discover in the process, by looking at the sequence a little more carefully, is that some of these amplified K3Ls have now picked up some mutations. And what's the... there's one mutation that shows up repeatedly.
Right.
Looking to see what it is here.
H47R.
H47R. Okay, so that's a particular amino acid change from a histidine to an arginine. And I think they even came up with a couple of viruses, right, that only had, that
Yes.
Yeah.
didn't have amplified K3L. They had only one copy.
Right.
But it contained this mutation. So they saw some viruses that had only one copy of K3L and had this mutation, and some that had multiple copies and had this mutation. And so the model becomes that what happens is that the viruses amplify K3L, you get a bunch of tandem repeats, and that allows them to grow by swamping out the PKR. And it also provides an opportunity, in my mind at least, to play with those.
Right, because you have extra copies.
You got extra copies. And experiment over time with mutations that can by themselves overcome PKR. And lo and behold, this one mutation comes up, which all by itself in a single copy of K3L is a better PKR inhibitor. And so now you don't need the multiple copies anymore, and it winds up collapsing. And basically, if you have a bunch of tandem repeats in the vaccinia genome, and this has been known for a while, they'll recombine with each other and spit out all the stuff in between if it's non-essential and collapse back down to a single gene. And this is why they call it an accordion. So it expands to give you more K3L, and then ultimately one of those copies gets mutated to this thing that works better against PKR, so you don't need the multiple copies anymore, and those all get recombined out, and it collapses back down to a virus that contains the single K3L mutation.
Right.
Fascinating.
Also, if you put the viruses with the amplified genomes in a cell where there's where the PKR isn't as inhibitory, it also collapses, right?
Right.
There's
Right.
no selection for it. Yeah.
Right. If you take away either part of the selective pressure, then it collapses back down.
So, Rich, this change H, 47R, can you tell us about the I word?
Oh, yeah. Somewhere. I actually
You wrote it out.
I wrote it out because I knew you. Remarkably, this exact substitution was previously identified in a genetic screen for K3L mutants with enhanced activity against human PKR using a yeast assay interrogating the K3L PKR interaction.
I bet the yeast assay didn't actually interrogate anything.
Don't think so?
No.
That's funny then. Is it both papers? The other papers? Yeah, both papers use the word now.
Yeah.
So, one of you guys, could you talk about the Red Queen hypothesis? I want to hear somebody wax philosophical on this, 'cause I get confused every time this comes up. What is the Red Queen hypothesis, and where does that come from?
If you have two organisms competing, or if you have an organism competing in an ecosystem, it's going to be competing with other organisms that can also evolve. And so it's going to have to adapt, and then something else will adapt to its adaptation, and it will have to adapt back, and so on. And you get this tit for tat. And you see it certainly in viral infections. The virus infects the cell. It develops a set of adaptations to get around the cell's defenses. The cell is going to have to deal with that by coming up with its own adaptations to get around the viral circumvention of them. So you get something like this PKR mechanism that's going to detect double-stranded RNA and shut things down, but then the virus is going to develop defenses against those, and this keeps going on. And I think it was Richard Dawkins who labeled this the Red Queen phenomenon or Red Queen hypothesis. And it's from a line in Lewis Carroll's Alice in Wonderland, which Vincent has pasted in the actual stanza here, but it takes all the running you can do to keep in the same place, is the Red Queen's line.
Actually, in Looking Glass. Alice and the Queen are talking, and Alice is saying, in our country, if you ran for a long time, you'd get somewhere. And the Queen says, well, here, it takes all the running you can do to keep in the same place.
Right.
So.
Dawkins was brilliant. What else did he do? He coined something else.
Selfish gene.
Selfish gene, thank you. Just talking about it the other day. So you get it, Rich? It really makes sense, you know? Both virus and parasite and host mutate, and you never get anywhere. Well, they all exist, I guess. So.
Well, and they do change. You know, you can have disruptions of the system that cause a new equilibrium to form, and that gives you that punctuated equilibrium that Stephen Jay Gould talked about. But those shift things to a new equilibrium where it still takes all the running you can do to keep in the same place at the new equilibrium until something else pushes it out.
I'm going to paste into the show notes a figure from the Malik lab. You can click on that, which they have modified to fit the red corn hypothesis.
Yes.
You see Alice being chased by viruses.
That's pretty cool. That could be an image for the episode.
I think that should be our episode image. Yeah, I think it's
so.
Really cool.
A couple of things come up with this. First of all, they ask the question: okay, so we forced the virus to do this. Is this something that really goes on? And so one thing they did was sort of take an unselected population of viruses and do mass sequencing on them and ask whether there are any duplications that go on in vaccinia in the normal course of events. And they can detect that, yes, this kind of stuff happens. And also from my own experience, and they quote some other papers out there, there are other DNA viruses that have evidence of multi-gene families in them, as if this sort of duplication can go on. Some pox viruses, but I think more notably some other herpes viruses. Now, one of the things that occurs to me in particular with pox viruses, and I want to ask you about this, Kathy, and we can consider other viruses. Pox viruses can accommodate a lot of extra DNA. So you can stick in at least 20 kilobases of DNA and without, at least in culture, impacting on the fitness of the virus. It replicates just as well. But in some other viruses, there are restrictions in how much DNA that you can add just because you can only stuff so much into the capsid. And so I'm imagining, for example, in adenovirus that there might be some restrictions on this. Do you have any thoughts on that, Kathy?
Right. And in fact, they comment on that, that adenoviruses and certain phages would probably not be able to do this amplification mechanism in order to generate mutations. Adenoviruses, at least for AD2 and AD5, I think this that we think of is that they can package about 105% of their genome size, 105, 106, something like that. So for a 36 kilobase genome, you can do the math. And that's because it's just got this fixed icosahedral capsid that's all the more space you have.
Some herpes viruses would seem to have the same sort of problem because they have a rigid capsid inside the envelope as well. And yet, if I'm not mistaken, there are some herpes viruses with significant evidence of multi-gene families. So I suppose it depends on the circumstances, because in some cases you might imagine you could duplicate some genes and lose some others to make room if under that particular circumstance you didn't need those other genes.
Now, the smaller DNA viruses are not likely to do this. The polyomas, the single-stranded DNA virus, circoviruses, right?
Right. I would imagine that even a bigger problem there, yes.
But the huge viruses, the mimes, probably do this all the time, right? In
Which
fact,
may actually be one of the reasons that they got to be that size.
Yeah, you may remember we did a paper a long time ago where they took one of these large meme-like viruses, and when they propagated in amoeba, you lose a lot of genes in the middle of the genome. You remember that? So that maybe those were acquired for multiplying in some other host, and then you stick them in amoeba in the lab, and they go away. Who knows?
Yeah.
It's also worth pointing out that this notion of gene duplication or amplification as an evolutionary process is has been out there for a long time in cells. One of the ways that tumor cells acquire resistance to drugs is to amplify drug resistance genes. And just in normal evolution, I think one of the classic examples is the beta globin locus. Okay, this is one of the subunits of our hemoglobin, and in humans we have I forget what now, 4 or 5 different flavors of beta globin that are expressed at different times during our development and are handy for having different affinities for oxygen that are appropriate for the different times during development. And theoretically, the way that evolved was from an original gene duplication or amplification, and then evolution of individual copies of that gene to acquire different functions. So this is a pretty common theme in biology, but it's really cool to see it happening in this particular circumstance with the poxviruses.
And they talk about how this may be a lot more common than we realize because people haven't been looking at copy number variation in viruses.
Right.
Typically, it's not really something that the sequencing technology was doing even just a few years ago, and it's not something that people have bothered to do. So they point out this interesting observation that that Variella minor and Variella major are less than 2% divergent at the genome sequence level. So, one's lethal, the other's not. Right. Is it just this 2% sequence divergence, or is there copy number variation involved in that as well?
Interesting.
Right. There's a nice preview or commentary in the same issue of Cell by John Roth and D.I. Anderson, and they talk about how this kind of amplification has also been shown to happen in bacteria for the lac operon and generating resistance to antibiotics and antipeptides. So, and they have references to a couple of really nice reviews if people want to follow that up even further.
Yeah, viruses don't invent anything; they just steal.
They probably invented a thing or two.
Yeah, well, yeah, maybe 1 or 2 things. Okay, I'll give that to you. Nice paper, though. Thanks for
nice
bringing
paper.
it to our attention. And I wonder what Eld Nels is doing next. Nels,
something cool.
Hopefully.
Yep.
So let me read the letter from Patrick that made us do this paper. Dear TWiV crew, after many episodes of listening, I thought some active participation was in order, especially after meeting Dr. Racaniello over drinks at the Brokosh Irish Pub during ASV in Madison. First, thanks to the entire TWiV crew for your service to the scientific and lay communities. I think that an enhanced science presence in the classroom, particularly early on, could really be transformative on many levels. More inquisitive minds, more kids amazed at the complexity of life, more logic-based thinking. The point I'm trying to get to is that TWiV is an awesome vehicle to disseminate science, and having done many hours of outreach in classrooms, it's very empowering to see how many people can be reached. So many thanks, and also a sincere offer to help out the TWiV cause in any way possible. I'll have to file that one away for easy finding.
And now, he just helped out the TWiV cause.
Yes.
Right. And now, tidbits from the random thought generator of my mind. Tidbit 1. In a TWiV covering an influenza virus in bats, there was some question as to the cycle of transmission and whether the virus could have come from insects. This was considered unlikely because insects do not harbor orthomyxoviruses, but in fact this is not true. There is a family of influenza-like orthomyxoviruses, thogotovirus, dorivirus, that are tick-borne infections of rodents. Interestingly, selection in insects has resulted in the glycoprotein of these viruses being more similar to baculoviruses than influenza viruses.
But ticks are not insects. Well, minor nitpick. Actually, I guess it's not a nitpick, it's a tick pick.
They're arthropods?
They are arthropods. But they are, what are they, arachnids?
Arachnids.
Yeah, this came up once before, and I had to look that up.
That's
right.
They're
arachnids.
They're arachnids.
Yeah. Thogotovirus.
Yes,
that's cool.
I'd forgotten about that, right. These are tick-borne viruses, and they are in the family orthomyxovirus. That's absolutely right. Tidbit 2, we had discussed an enhanced multimedia interface for ASV. I think that CROI, Conference on Retroviruses and Opportunistic Infections, does this really well, and the fact that all the talks are free and online hasn't stopped people from going to CROI. Yeah, we talked about doing that for ASV, and I think this is a good argument that a lot of people at ASV are worried that if you broadcast the talks or some of the talks, people won't go to the meeting. But I don't think that's correct.
That is absolutely false.
Yeah, I agree. And CROI is a good example.
I do meeting coverage as one of my regular gigs for different organizations, and when they webcast a meeting or put the meeting online, there are plenty of people at the conference. People want to be at the conference because you get the interaction that's there that you're not going to get in a web conference. That's absolutely not a danger.
So I've been talking with Barbara Sherry, who's the president of ASV for the next year. We're trying to move ASV into the new media age. And I had suggested to webcast or at least put online some of the talks. And she had said that there is resistance because of that, but I will try and use CROI as a persuasive argument. I suggested they just try it.
This looks like a good model for this.
So given the conservative nature of the ASV management, I said, look, take one talk, take the keynote, record it, and put it online, and do an experiment. See if it affects attendance, and I bet it won't.
Right. And ultimately, eventually, they produce the DVDs of the talks. And from the business meeting, I seem to recall that was not a moneymaker.
No, they lose money on those, yeah.
And so this might be a more economical way to propagate the same thing. I mean, I assume that once they're webcast, they're up there forever.
They can be. They don't have to be.
I see.
But I think they should put them online for people who don't mind. You get their agreement to do it. I think it would make people go to the meetings because they say, oh, cool, this is a great meeting. I want to go to it.
And you don't have to live stream it.
No, you don't have to do that. You just record it.
I know a bunch of meetings have tried that, and they usually end up saying, most people watch it later anyway.
Well, CROI live streams and records.
Right.
And so does ASM. But I agree. You don't have to. You could just, they have the video on the DVD. They can just put it online. So
yeah. I would get somebody from CROI who does this and knows about it to come to one of the ASV council meetings and give a presentation on this.
And by the way, if they do this sort of presentation, another thing that sometimes a lot of people appreciate is a synopsis of the conference that they can read as a shorter version of watching the talks,
right.
Which is actually one of my main lines of work.
Say, is that what Alan Dove does?
Yes, yeah. just a little plug there.
Tidbit 3, although I hope this is of general interest to the entire TWiV crew, I thought that Rich in particular would find this interesting and perhaps might result in a TWiV bump for new faculty Nels Eld, lead author, and Harmit, senior author and my mentor. And that, of course, is the paper we just did. So thanks for that, Patrick. Hope this finds you all well, and thanks again for TWiV. Patrick, it's 26 Celsius and absolutely beautiful in Seattle. All right.
Cool.
The next one is a link from Ricardo, who links to a book, one of the new iBooks, by Carl Zimmer and Douglas Emlen, called Evolution Making Sense of Life. Science writer Carl Zimmer and evolutionary biologist Douglas Emlen have teamed up to write a textbook intended for biology majors that will inspire students while delivering a solid foundation in evolutionary biology. It is free, so if you have an iPad, you can just download this and read it for free. And there are 2 reviews here, which are highly contrasting.
Yeah.
So check it out. And then let's see, Alan, take the next email, please.
Sure. Dear Twivtians. Twivtians?
Twivtians, like Martians.
Twivtians, like Martians, yes. Your delightful podcast, this is from Kimon, or Kimon?
Or Kimon.
Kimon? I'm just screwing this thing up. I haven't even started the letter. All right. Your delightful podcast has helped to steer my interests towards virology and immunology. I studied mathematics as an undergraduate and realized that my calling was in medicine while completing the Teach for America program in New Orleans. I will be starting medical school next month at the University of Pennsylvania. Congratulations. And I'm looking for particular directions of research that might be appropriate for my quantitative background. I'm particularly interested in HIV/AIDS, but very flexible given that I'm just starting out in the biomedical research world. Any suggestions of papers, books, people, or journals that I could seek out would be incredibly helpful as I try to understand where my skills as a math guy would be most useful. Thank you for sharing your conversations with the world. Your work has had a measurable positive impact on my life. One word for you: bioinformatics.
I was going to say exactly the same thing.
Anybody want to say plastics?
Yeah.
I was thinking the same thing. Right.
Rich
Yeah.
is a
Go look
plastics
at
guy.
go watch the movie The Graduate.
Yeah. Kathy?
The other thing is to think about mathematical modeling. So people
Yes.
that are, modeling the immune system, there's people in my department that do that. And so having really strong mathematics skills is very useful there as well. So.
Yeah, there's lots of opportunities for a math guy.
And probably just your average bench lab, if you get turned on by what they're doing, your quantitative skills will come into play, and the way that you reason and think will affect how you do research. So basically almost anything.
Yeah.
Basically, we are swimming in nucleic acid sequence data, and we don't know how to deal with it. We don't know how to sort it. We don't know how to store it. We don't know how to interpret it. And it's going to take math guys to do that. And ultimately, the model, it morphs into modeling as well, because, I think one of the cool areas is if you have a primary sequence of a protein predicting the tertiary structure and function from that. There's also epidemiological modeling as well, so there's all kinds of stuff to do.
Lots of data you can interrogate.
Yeah.
I'll have to edit that out.
Or you could just analyze it.
So is interrogate now a dirty word?
No, it's okay, but I'm just going to complain whenever I hear it, unless I hear a good reason that it's right. Rich is a guy here who developed a program to predict protein-protein interactions, and it's pretty good. People have tested the output, and it's pretty amazing that you could do that, right?
Wow,
I mean, specific, not just general interaction, very specific. You take a protein, you put it against a database, you scan the database with the program, and then you can find interactors.
Wow.
Rich, can you take the next one, please?
Okay. Rohit writes, Dear TWiV doctors, I was just listening to the TWiV 190 where Dr. Racaniello was talking about making TWiV accessible to more people, especially in the developing world where people do not have regular access to internet. I think radio can be a very effective way of spreading scientific knowledge not only in parts of Asia and Africa, but also right here in the United States. Have you explored the possibility of using local radio stations such as NPR in the United States for increasing the audience of TWiV and the other two podcasts? I'm not sure how much the radio station administrators will be interested, but it is definitely an area worth exploring further. Maybe TWIM already has a listener who knows more about making it happening. As always, love the podcast. Keep them coming. Thanks, Rohit. Sure, I'd love to be on NPR.
Sure, it'd be awesome to be on NPR. I think there's a big bandwidth problem there, though.
I do know of one thing that started out, I believe, as a podcast and is now carried at some NPR stations, and that's a show called Ask Me Another. And so I think they were doing podcasts in somewhere around New York City, Brooklyn maybe, and now at least our NPR station carries them on Saturday mornings.
How long are they?
It's an hour.
Yeah, we'd have to cut to an hour. If
we'd have to stick to a time limit, certainly. And there's also the issue that there's a limited number of radio channels. There's an infinite number of internet channels. Well, finite but very large. So given the limited bandwidth on radio, you have to have a large enough audience for the show. And while our audience is large and growing by podcast standards, it's not really up to the point of what radio needs to make it work.
Yeah, they have high costs to maintain that infrastructure, so they have to pay it.
Car Talk is going offline. Those guys are done. They need a niche there.
Yeah, more people like cars than viruses, though.
And didn't somebody refer to TWiV as car talk for viruses?
Yeah.
Yes.
So.
Well, I don't know how to even approach this, and it's way above my pay grade, but if anybody's listening...
I think that stuff, if anybody's listening, that stuff happens.
It might just happen. We got a mention on NPR back in February, after doing the TWiV in Dublin, so that helps. I figure we just hammer away, and eventually we'll get discovered, right?
Eventually they will be unable to avoid us.
That's right. Rohit, by the way, is a postdoc. I met him at ASV.
I did too.
In Kartik Chandran's lab, which is at Einstein, and we did a paper of his some time ago on the Ebola receptor. Cool stuff. Kathy.
Okay, Jim writes, I wanted to make sure you didn't miss this news item. And it's this is a couple weeks ago now that I looked at this. It's a show, and it's got 5 different parts in it. But one of the items is the 4th item, concerns the very smart idea of using cell phone tower power systems in developing countries to reliably run refrigerators used to store vaccines. And so then there's a link to that, and I think I provided a link that goes right to the story from PRI's The World. Anyway, so yeah, it's about keeping vaccines cold using cell phone tower power, and the fact that cell phone towers are becoming more ubiquitous leads to you would then be able to maintain the cold chain for those vaccines that need it.
It's a great idea.
I wonder if the cell providers would like that, though.
If you pay them.
Yeah.
Sure.
Maybe if they get something out of it, absolutely.
Maybe they'll even be charitable and give you a cut rate.
What
fascinates me is that the motivation of telephones is stronger than the motivations of keeping people healthy, right? So it's easier to maintain a cold chain if you piggyback it on telephones than it is just to set up the cold chain to start with.
Yeah.
Well, it's because public health is fundamentally negatively based. When it works perfectly, nothing happens. Whereas cell phones are an immediate technology that allow you, particularly in third world countries, they're an incredibly powerful tool. Suddenly you can communicate with somebody elsewhere in the world. So it's an immediate and easy to appreciate technology, whereas vaccines are a little more abstract.
Right.
Communicate and play Angry Birds.
Yes, very importantly.
They're never going to put wires there, so this is really important, too.
Right.
I think it's a cool idea. I think we ought to move on to picks. What do you think? It's at an hour and a half now.
Sounds good.
All right, save these for next time. Kathy, what do you have?
So I picked 2 links to Vi Hart. She's this amazing mathemusician. She has a very large collection of things on her website, including math doodling, math food. Balloon twisting is the one that caught my eye this time. She has a way that you can twist balloons and make an icosahedron. Looks very virus-like. So you could lose yourself for a long time on her web pages and just be fascinated.
Wow, a lot of stuff she does.
Yeah, very cool.
Well, Kathy, you need to do this for virology.
To to twist the balloons? You mean?
Yeah, just.
Oh, you mean make a website for virology?
Yeah,
like she's got.
She's a mathematician. You could be a viral musician. Are you a musician?
I am a musician, but this is way beyond what I could do.
Okay.
What do you play?
I play handbells. I play recorder, and I sing.
Cool.
You should be in Herpetic Legion.
That was an interesting band at ASV. Yes.
It's I recorded it. It's up on the web. I don't know if you know that.
Okay.
The video is up on YouTube.
Check it out.
youtube.com/PROFVRR. Alan, what do you have?
I have speaking of YouTube, I have a YouTube video from the New England Journal of Medicine, which recently, I think, turned 200 years old or something like that. And they have done this nice little, neatly produced short documentary called 200 Years of Medicine, Getting Better. And it's they focus on a few different subjects. They've got a segment on surgery, they've got a segment on oncology, and they have a segment on infectious disease. You'll see some people who you probably recognize in the video.
You mean
and it's just
you're in it?
No, I'm not. But you probably recognize Tony Fauci. He shows up. A couple other folks.
Tony.
So they have interviews with these people and some clips of what's going on in the hospitals and how this has changed and how each field has been affected differently by different types of progress. Surgery, it was as simple as washing your hands. It was a huge advance, and anesthesia. And then in infectious disease, the focus is on HIV, where it happens in modern times, and the history is very compressed. So it's just a neat little short documentary. I think it's 15 minutes long or something.
It says 44 minutes here.
Oh, 44. Oh, I'm sorry. It's not that short a documentary, I guess.
Yeah,
it seemed to pass quickly.
The cool thing is you can just pass your cursor along the scrub bar and get...
You can fast forward, yes.
Get little thumbnails without actually playing the video. You can just see thumbnails at different parts. I just discovered that. Did you know that you could do that?
I think that's probably new. Yes, I just discovered it too. I had not seen that before. I think that's a new feature.
It is pretty recent, I think, yeah.
This looks really cool.
Nice. 1812. All right, thank you.
Sure.
Rich.
My pick is a spin-off from Kathy's pick, because I was looking through Kathy's pick, and Vi said that she was now an instructor at the Khan Academy. And I thought to myself, what is the Khan Academy? I had never heard of this before. And
it has nothing to do with Star Trek.
I looked up the Khan Academy, and I got completely lost. This is and the link I have here, I think, is a link to a TED Talk by the founder of the Khan Academy, Khan himself, which is embedded into the Khan Academy site. So you could start there and watch the video and then cruise around through the site. But basically, this is not-for-profit, free for the consumer, online education. And it starts out, this guy was a financial analyst for a hedge fund in Boston, and he was tutoring his nieces. And the way he was tutoring them was to make little videos. I think it was started off in math, tutoring them in math. He'd make little videos, and he posted them on YouTube because it was convenient. He discovered his nieces liked the videos better than they liked him, among other things, because they could stop him and rewatch him and all that kind of stuff. And so he just kind of kept going, and this snowballed into this Khan Academy, where they make online educational materials, and it's become, there's a variety of these things now. It's become pretty sophisticated. They're actually testing it out as a supplement to textbooks in the school systems in Palo Alto. So I thought that this was at least interesting and potentially a model of what the future might be with the internet in the classroom. I thought it was really great.
Cool.
Yeah, Khan gets big bucks from...
Yes, Bill Gates.
Bill Gates, yeah.
And Google.
I want Bill Gates to discover us.
Yeah.
Well, I think that's what as I was looking through this and I realized that they got grants from both Google and Bill Gates, that made me sit up a little straighter and say, I need to dig into this, as somebody is taking these guys seriously. Mm-hmm. And some of the stuff his TED Talk is great, and some of the stuff he talks about is interesting. As a sort of a control, I went and looked at a video that he's got on viruses, and that he made himself. He's turning himself into a Renaissance man with this because, like I said, he's a financial analyst for a hedge fund originally. And his lecture on viruses, it has, a couple of things that I might question a little bit, but for the most part, he got it right, and it would be very digestible for somebody who has never thought about viruses before. So not bad.
Cool.
Not bad at all.
Yeah, I think some of our listeners have mentioned this in the past, but always good to re-mention. All right, my pick is a little iPhone app. Little because it's cheap, I guess. They're all the same size, more or less. Well, maybe not. That's not true. Anyway, it's called Tweet Keeper. And if you use Twitter, this basically will grab all of your tweets. So Twitter only lets you retrieve 3,200 tweets. And if you use this thing, it pulls them all off, and then every time you launch the app, it just pulls your recent ones. So you can basically have all your tweets on a text file. All right. Now, I only have like 2,200 tweets. So I just downloaded them here. Now, you may say, what the hell do you need all your tweets for? Well, at ASV,
That's exactly what I was about to say.
In fact, I don't need them all, but the ones I did at ASV this summer, I live tweeted many of the talks, all right? I wanted to put them on a website for people. Many people said, I'd like to see them all. And there's this other service called Storify.
Storify, yeah.
I tried it. It absolutely is horrible. It's full of bugs. It doesn't work. It crashes. It loses file. I spent hours wasting time. This thing, I get all my tweets. I can do what I want with them. So I did that, and it's funny, my first tweet, I got to pull this up, because this shows how Twitter has evolved. None of you guys are tweeters, I know that, so I'm I'm talking into the...
I was on, and then I was off, and now I'm back on again.
My first tweet, when I first doing Twitter...
Is your first ever tweet?
Yeah, the first one.
Wow.
Where the hell is it?
Historic.
So I have a record on my iPhone of all the tweets I ever did. Anyway, my first tweet was... over 5 years ago, I think. And it was basically like, I'm on my way to a faculty meeting. And that's why Twitter is crappy, because nobody cares what you're doing. But within a year, I had morphed into telling people things, which is what the good part of Twitter is. Mm-hmm. Twitter is. So if you do a lot of tweeting and you want to save them, this is really cool, because this will save everything. And now I can do a live tweeting summary from ASV, which I'll probably never get to because I don't have time. But anyway, it's a cool app. It's $1.99. Check it out.
Cool.
What else have we got here? That's it. Oh, we have a listener pick of the week. We have two. Yeah. One is from Diane. Hi, TWivers. Thanks, as always, for an excellent podcast, as well as TWIM and TWIP. I especially loved the How to Read a Study Helpful Hints segments, as it was really nice to hear that learning to read studies is a skill that takes practice and education. And to know that I will have to struggle with reading studies for a while before I'll be really able to parse them. Anyway, I recently read a young adult book called The Way We Fall by Megan Crew. It's a story about an island in eastern Canada, if I remember correctly, where a viral pandemic hits. The fatality rate is high, and the island gets quarantined. I don't know how accurate the science is, but I thought it was a fun read. Amazon says this is part one of a trilogy, so I guess there's more, but at the time I finished it, it felt like an actual ending. Not great literature, but a fun, gripping, and quick read. Keep up the great podcasting. I love listening and learning. The Way We Fall. Look at this. Magically, a link has appeared.
Yes.
Yeah, it is a trilogy. Okay. I'll have to check it out. If it's a quick read, no problem. Stephen sent us a link to an online bulletin board community dedicated to photomicrography, amateur microscopy, and photomicrography, called amateurmicrography.net. I can't say that properly.
Amateurmicrography.net.
It sounds weird, doesn't it?
Micrography, yeah.
Anyway, it's pretty cool. It's got a lot of neat galleries in it. And Stephen writes, it's 9 p.m. and still 32 degrees centigrade.
Ooh.
Yeah, it's pretty warm. Summer does that. And that's it. Anybody want to say anything else?
No.
Okay.
Nope.
TWiV is at iTunes, the Zune Marketplace, and TWiV.tv. And if you're new to TWiV, you should subscribe in iTunes and leave a comment or a rating. That helps us to stay very visible over there. It's pretty crowded on iTunes. And that way more people can discover us. We'll get lots of listeners, and NPR will come a-knocking, as well as Bill Gates and Google. We're just waiting here, waiting in my ivory tower. Also, we have a Facebook page, facebook.com/thisweekinvirology, where at the moment there are 2,600 people checking us out. We also have an app for your iPhone or Android device over at microbeworld.org. And finally, do send us your questions and comments to TWiV at twiv.tv. Kathy Spindler is at the University of Michigan, which is in Ann Arbor, in some part of Michigan, 6 to 8 hours from Madison. Chicago, is it, or Madison?
From Madison.
6 to 8 from Madison, 4 hours from Chicago, right?
Thank you, Kathy.
You're welcome. It's fun to be here.
Rich Condit is far away from Ann Arbor. He's at the University of Florida, Gainesville. Thank you, Rich.
You're welcome. It's raining.
Are you sitting next to your window there in your office?
Oh, yeah.
Well, aren't you getting the edge of a hurricane at the moment?
Yeah, that's right.
Well, not really. That kind of passes over. It was really disappointing.
Really?
Yeah, it just really didn't. It really didn't do much at all. We got about an inch of rain, and that was it. So this is just sort of Florida rain. I haven't actually looked at the radar, so I don't know where this is coming from. But the hurricane is kind of long gone.
Right.
By the way, Vincent, are you going to tell our listeners that we aren't going to be around next week? Because if there's a blank spot and they don't know about it, there's people out there are going to be scared.
I will.
We're in withdrawal.
I will.
Yeah.
I will. Alan Dove is at alandove.com and on Twitter as Alan Dove. Thank you, Alan.
Always a pleasure.
And I'm Vincent Racaniello. You can find me at my blog, which is virology.ws. So as Rich said, we will not have an episode next week. And the reason is that we have to get number 200 for where we're going to be on a certain date. So if we did one next week, we would be 201 at this special place where we're going to be. We don't want to tell you because we want to make a really big surprise. So that's why we have to skip one. How's that, Rich?
That's good.
But the thing is, nobody listens to the end. So no one will know. Because
nobody listens to the end of the podcast?
No, nobody. Very few. Kathy Spindler is the only person in the world that I know.
Is that right?
Yeah.
Well, they're really missing it because there's the outtakes occasionally and all that stuff.
Exactly.
After dark, Easter eggs, all that stuff. Yeah.
Yeah.
Because last time we skipped an episode in July, I think, this year. And on Monday, everybody was tweeting and saying, "Where's the episode?" I said, "Hey, listen to the end of it. It told you there wasn't going to be one." So I guess we should have said it at the beginning, but
tough. You got to listen to the end. If you don't listen to the end, it's your own fault.
You got to watch all the movie credits too. Sometimes you're entertained.
Oh, I love doing that. Yeah, I'm always under pressure to leave though. But after watching Toy Story, years ago, I learned to sit for the credits. Yeah.
Right.
Did you sit for the credits in what is it, Iron Man? Did you sit for the credits there?
I think so.
Yeah.
Because that was very weird. Didn't make any sense. But anyway. So, yes, we're not going to be here next week, but no one will know. So I'm speaking in a forest where there's no noise. It's okay. If any of our listeners are in Nebraska, there is this virology symposium on October 5th, which I'm speaking at along with Grant McFadden and the guy in Vanderbilt whose name
Terry
Dermody and one other person. And then
so now you're making an announcement that is only for fans in Nebraska who listen to the end of the show.
Yeah, there's 0, so I shouldn't even bother, right?
All right, so both of you listeners in Nebraska, besides the symposium, we're going to do a TWiV there, so check it out. I'll put a link to a poster. There you go. I could put this at the beginning, but I don't want to bother doing it. It doesn't fit, But we should learn to make important announcements at the beginning, I agree, but not this time. All right. And now for the part that no one except Kathy listens to. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.